Hello Year 8, this is now lesson 4 in the Food Glorious Food unit. We're going to start this lesson with a little quiz just to recall the information that you've learned in the previous lessons. So I'd like you to answer these questions either on the back of your booklet or on a piece of plain paper. Okay, so you've got 5 questions to answer. Pause the video now and get your answers written down, please. Okay, so let's have a look at the answers to these questions. So if you check yours as we go along. Question 1, which food group is required for growth and repair? Would be protein. A lack of vitamin C can lead to which disease is scurvy? Question 3, which food group do potatoes and pasta contain a lot of? Is carbohydrate. Question 4, a shortage of vitamin D leads to which disease? Rickets. And finally, question 5, which food group helps food to pass through your digestive system? Is fibre. At the end of lesson 3, you were asked to complete homework 2. So the next thing we're going to do today is just to go through those answers and make sure that you've got a mark for that homework. Obviously, if you've done this on the online activity, you've already got your mark and you can just fast forward through this part of the video. So if we look at the first one, dear Mum and Dad, I am finding life as a student tough. I miss your meals and I'm spending every night down the chippy and have put on some weight. Love your only child. PS, any chance of a loan? This one should be joined together with I eat too much oily and fatty food and do hardly any exercise. Dear historian, I am writing a project on sailors in the 16th century. Can you tell me why so many of them suffered from scurvy? Yours, project writer. So obviously this is something we've looked at over the last couple of lessons. Scurvy is caused by a lack of vitamin C. So this one goes with this one here. Very little fresh fruit and vegetables were available to them, so they often lacked vitamin C. Number 3, Doctor, I am finding it difficult to do number 2s. I am always constipated. What can the matter be? Awaiting your answer urgently. Yours, uncomfortable. So a lack of fibre can lead to constipation. So this person needs to make sure that they are eating cereal once a day and plenty of vegetables to get enough fibre. Dear Doris, can you help me? My child does not seem to be growing. Can you tell me what could be wrong? Mrs. Anxious. So as we've looked at previously, for growth you need protein. So this one should be joined up with the top one. I need to eat more meat, fish, cheese and milk. All of these will provide me with lots of protein. Dear coach, I believe that I should be picked for the team as I train regularly and have been on a special diet to increase my energy levels this season. Yours faithfully, Fit Man. So for energy, Fit Man will probably have been eating a lot of carbohydrate. So it's joined together with this one. I eat a lot of carbohydrate-rich food like bread, pasta and rice. The last one, dear teacher, my son has broken his arm falling off his bicycle. The doctor has said he has weak bones. He will not be in school this week. Mrs. a worried parent. There's obviously only one left. For strong bones, we need calcium. So I need to drink more milk and dairy products to increase my calcium intake. Can you now write your mark out of 6 at the bottom of the page, so that's page 11, but also onto the front of your booklet in the table. So what we're going to do in today's lesson is to have a look at how we would test different foods to find out if they contain these food groups that we've been talking about. So if I was to give you a sample of a food, you would be able to identify whether it contains protein, whether it contains fat, whether it contains carbohydrates. So with carbohydrates, there's actually 2 carbohydrates that we need to be able to test for. As you know, carbohydrates are split into the starchy foods and then also the sugary foods. So we're going to start by testing for starch. Starch is a carbohydrate made by plants when they carry out photosynthesis. So they make glucose first of all, which is then combined together into starch molecules to be stored as a store of energy. So I'm going to show you a video now of how we would use iodine solution, as you can see on the screen, and this is on page 12 in your booklet. If you can make sure you're putting the date at the top of the page. And we will now find out how you would test for starch using iodine solution. Okay, so to test for starch, we've got there a bottle of iodine solution. As you can see, it's sort of an orangey colour. In the first tube, which says C, I've just put water. So this will show you what happens to iodine if it's just put into water. As you can see, it's staying the same colour. So it's still that sort of orange yellow colour. In the second tube then, we have starch solution. So when we add the iodine into the starch solution, as you can see, we've got a very clear colour change there. So the solution turns very sort of dark blue colour. We would refer to that as blue-black. So that is your positive test to see if something contains starch. So in your booklet on page 12, we're going to now colour in the diagram that you can see on the board. So I'd like you to get an orange coloured pencil, and we're going to colour in the bottle of iodine solution, okay, to show what colour iodine is. Obviously you can make yours a bit neater than mine. If you need to pause the video at any point, please do. So when the starch is not present, what we saw was that the iodine solution stays the same colour, so we don't get a colour change. Whereas when starch is present, we get a distinct colour change, and it changes to blue-black. So we're going to label that then on page 12 as blue-black. So if you can make sure that your diagram on page 12 now matches up with what you can see there on the screen. So the second test that we're going to do is for glucose. So this is the other type of carbohydrate which we often find in foods. So glucose is a simple sugar. So to test for glucose, again, I'm going to show you a video of how we would do that using Benedict's solution. So again, with this, we've now got some glucose solution in one tube, and in the first tube with the C, we just have water, again to show this difference in the colour when glucose is present. Okay, so the first tube has water, the second tube has the glucose solution. So to test for glucose, we would use Benedict's solution, which is a blue coloured liquid, as you can see there on the screen. So if we add the Benedict's first of all into the water, you can see that it's staying blue. But you'll also see that in the glucose, the Benedict's has stayed blue as well. So we're not seeing there any colour change. This is because for the colour change to happen, we have to provide the solution with heat. So we're going to put the tubes now into a beaker of boiling water, and that will then cause the reaction between the Benedict's and the glucose to happen. Okay, if you were doing this in the lab, you'd probably use a Bunsen burner to heat it up, but boiling water from a kettle works just as well. Okay, so we've taken the test tube rack away so you can see this more clearly. In the tube with the glucose, you can see it started going sort of a green colour first of all, but then we're getting a colour change at the top into sort of an orange colour. It does take a couple of minutes for this colour change to occur. So we've speeded up the video a little bit, and this is the final colour that you would be looking for. I always think it looks a bit like tomato soup, but what we'd refer to that to is brick red. So the colour change with Benedict's when heated up is brick red. So we need to go back now to page 12, and we're going to colour in this picture. So we'll start with the bottle of Benedict's. If you remember from the video, Benedict's is blue. So if you can just colour that solution in here. Okay, so we've then got glucose in the tube, which was a clear solution, and we would have the Benedict's in the pipette. You could even put a couple of drops there to show that it's being added into the glucose. And then finally, I know it's slightly darker colour, but I'm a bit limited on the colours that I've got available to me. We're going to colour in, once it's been heated up, it turns that brick red colour. You will get different sort of shades, different darknesses of this colour anyway, because it depends how much glucose is in the solution. The more glucose there is, the darker it will turn. So we're going to label this then as brick red. So if you get asked what colour does Benedict's turn when it's added into glucose, it turns brick red. Right, so we're now going to go on to the third food test, which would be for protein. This is on page 13 in your book. So perhaps protein, the Biret test. As you know, proteins are used for growth and repair. So I'd like you to just pause the video and have a go at answering the question you can see there on the screen. Think about, what do babies drink a lot of, and can you give a reason why? For this question then, hopefully you've remembered that babies drink a lot of milk. Why do they drink a lot of milk? It contains protein. There's a lot of protein in milk. And obviously babies need to grow. They grow very, very quickly in their first year. So lots of protein is needed to help them do that. So milk contains lots of protein to help them grow. We're now going to watch the video to see how you would test for protein. So to test for protein, again, we've got a control, which is water, and then we've got protein in the second tube. We add in Biret solution, which is blue at the start. So as you can see in the water, it remains blue. And then in the second tube, when we add the Biret solution, we can see a colour change into a purple colour. We'd usually refer to that as lilac. So you can see there the colour change that we'd get if protein is present. On page 13 then, we're going to colour in our picture again. So if we can colour in the Biret solution. Again, make sure yours is a bit neater than mine. Just doing this very quickly. And then in the tubes, protein not present, stays the same colour. If protein is present, we saw that colour change to lilac. Obviously my purple's a bit darker. That's the best I've got. So we're going to label that as lilac. So you can now make sure that your picture on page 13 matches up with what you can see there on the screen. Obviously, if you don't have any coloured pencils at home, just label the different colours, and when you're in school, we can colour them in. The next test that we're going to do is for fat. Some fats are good for you. Obviously, too much fat in the diet is not and can lead to obesity, but some fats are good for you. They help to keep you warm. They keep some animals buoyant, okay, helping them to float in the water. They're used as an energy store, so if your body hasn't got a ready supply of carbohydrate available, you will start to break down fat and use that for energy. However, as we said, too much fat can be bad for you, okay, contributing to conditions like heart disease. So to test for fat, slightly different to the others, we're not using a liquid to test for fat, we're using filter paper. So again, I'm going to show you a video, and this is on page 13 in the booklet. So with fat, again, we're going to do a control. We've got a piece of filter paper there where we've just put a couple of drops of water, and we've got a piece of filter paper there where we've dropped some oil onto it. We would leave that to dry. Once it's dried out, the control with the water, you won't be able to see sort of where the water was. But as you can see, as the fat's dried, it's left behind a spot. And if we were to hold that spot up to the light, we would see that it is what we would refer to as translucent. Okay, so when we add fat to filter paper, we get a translucent spot. So translucent allows light through, but you're not able to see through it. So think about something like your bathroom window. That would be referred to as translucent because it's letting light through, but obviously people can't see through your window into your bathroom. On page 13 then, you've got some lines there where it says what you see. So on there we're going to say that the filter paper becomes translucent. Okay, be careful with your spelling there. Make sure that you've copied that down correctly from the screen. So the filter paper becomes translucent. Okay, so the last chemical test now that we're going to do is for vitamin C. So as we've discussed a number of times, vitamin C is a vitamin which if you are deficient in, you can get a disease called scurvy. It's really important in the body because it helps to heal wounds and gives you strong skin and capillaries, which are the little tiny blood vessels which are carrying blood and all of the supplies that you need to your cells. So why did many sailors years ago suffer from this? This should be an easy question for you to answer. So if you pause the video now and write your answer down, please. So what you need to say there is that on long journeys, they didn't have a supply of fresh fruit and vegetables. Okay, so on long journeys, they didn't have a supply of fresh fruit and vegetables. If you can make sure you've written your answer in on that line. So again, we're going to watch a video now with the test for vitamin C. So we're using a chemical here called DC-PIP. So again, now for vitamin C, we've got a control tube with water in and a tube with vitamin C in. We're going to use a chemical called DC-PIP, which is a very dark blue colour. So when I add the DC-PIP into the water, and with this one we only add 1 drop at a time. So you can see there that in the water you can still see the blue colour there of the DC pip. If we add 1 drop of DC pip then into the vitamin C solution, okay, you can see the colour in the pipette, but once the DC pip hits the vitamin C, the colour disappears. So that's our positive result. Once DC pip is in contact with vitamin C, it decolorizes, it becomes colourless. Whereas in the water, you can still clearly see the colour of the DC pip. So on page 14 now in your booklets, we're going to colour in the DC pip. So that very sort of dark blue colour. So make sure that you colour yours in fully. So in the pipette, the DC pip was still that dark blue colour. And we'll draw a drop in here, going into the liquid. But as we saw in the video, the DC pip becomes colourless. So it decolorizes, becomes colourless. That is then the positive test for vitamin C. And you can actually do this test to quantify how much vitamin C is there. The more DC pip that becomes colourless, the more vitamin C that there was in the solution. So if you wanted to compare different foods to see how much vitamin C was in there, you'd be able to do that using DC pip. Usually what we would do now if you were in the classroom was I would provide you with some different samples of foods, and you would choose 4 foods from the selection and carry out all of the food tests on them to identify if they contain a particular food group. Obviously, we're not going to be able to do that at this point. So what I would like you to do is to choose 4 different foods and then just do a little bit of research online to find out if they contain starch, glucose, protein, fat and vitamin C. So for example, in the classroom, usually I would give you things like bread, cheese, maybe some milk, some orange juice, so foods like that. So have a think about which foods you would like to find out. Write the name of the food into the first column and then just do a little bit of research to find out whether they would contain these different food groups. So pause the video and get that activity done, please. Lastly then for today, by the next lesson that you have, I would like you to complete homework 3. Now, I just want to make sure that everyone's clear on what they need to do on homework 3, because people often don't read all of the instructions and only end up with half the marks. So you have to follow these instructions carefully. So firstly, colour each pair in the same colour with a pencil. So find the nutrient and the chemical that you would use to test it and colour in the boxes in the same colour. So for example, if you think that we use DC pip to test for starch, you would colour in those 2 boxes the same colour. Obviously we don't. You should be able to identify what we do use to test for starch. You then need to join the pairs up by drawing a line between them. So the ones that you've coloured in the same colour, you need to join with a line. So that's for the first 5 marks. The bit that everyone forgets to do, which is for the second 5 marks, is the bit at the bottom. On each line, write down either the colour change or what happens during the test. So from what you've just seen in the videos, what colour change is happening or what is the positive result that we are looking for. Any questions, please get in contact with your teacher. That brings us up now then to the end of lesson 4. Thank you.